Hello, guys. Good day. Welcome to another topic for virology. For this video, we have influenza virus. So, for the lesson outcomes or learning outcomes, so we have the following. So, basically, it's the same as the other lecture outcomes that we had for virology, especially those focusing on specific families of viruses. So at the end, the students are expected to be able to discuss the viruses in terms of their classification and structure, in terms of their pathogenesis, their clinical manifestations and complications, and of course, laboratory diagnosis and identification methods, and if applicable, treatment, prevention, and control of the viral infections. So for Okay, so influenza viruses are part of the Orthomyxoviridae family of viruses. They're, of course, enveloped with RNA in their genome. They can cause contagious respiratory illnesses, usually affecting the nose, the throat, and the lungs. So technically, upper respiratory tract. However, they can cause lower respiratory tract infections and other disseminated forms as well. It can occur sporadically. It can occur as a pandemic and, of course, pandemic. So again, to recap, the important properties of orthomyxoviruses. So you can find this in your JAWETS, table 39-1. So their virion is typically spherical in shape, pleomorphic because of the envelope. They tend to change in form or shape. Their composition again is RNA, and their genome is single-stranded RNA and segmented into 8 molecules. So later, we will discuss the significance of its segmented genome. Their genome is also in the negative sense. And they do have structural proteins similar to the other infectious agents. Their envelope contains glycoproteins, hemagglutinin, and neuraminidase, which plays a very important role in their in their invasiveness as well as infectivity. And then these characteristics are actually basic to or common to the influenza virus type A. So Okay, so for the influenza virus classification, there are 3 immunologic types of the influenza viruses. So we have type A, type B, type C. No, compared to the other two, type A undergoes antigenic changes continuously. Okay, for type B, it does undergo antigenic changes, but not as common as in type A. Okay, and type C appears to be antigenically stable, so it does not show antigenic changes. Okay, now the antigenic changes or antigenic differences is usually based on the differences of the internal structural proteins, the nucleocapsid proteins, and the matrix proteins. Okay, so those 3 structures are responsible for identifying, or they are basically used as the criteria for differentiating the 3 immunologic types of influenza viruses. Again, that's internal structural proteins, nucleocapsid proteins, and matrix proteins. Okay, so they are the basis for dividing influenza viruses into the 3 immunologic types. Okay, now, antigenic variations in the surface glycoproteins, particularly the HA or hemagglutinin and NA or neuraminidase. In some books, they just write H for hemagglutinin, N for neuraminidase. Either way, they're correct. Okay, so they are used. So the differences in those glycoproteins, no, your H and N or HA and NA, are used to subtype the viruses. Now, the good thing about it is that only the type A is divided or designated into subtypes. So we don't subtype group B. Okay. Okay. So there are 18 different hemagglutinin types and 11 neuraminidase subtypes. Okay. So that's plenty. And imagine different combinations that you have, okay, that you can get from the 18 HA and 11 NA. Okay. Now, influenza type A strains are also known to be found in aquatic birds, in ducks, in chicken, also in pigs, horses, and in some cases even in seals. Okay. Now, it is only influenza A that is known to cause flu pandemics. Okay. It has not yet been known if type B can cause pandemics, but as the case is, as the case may be, it's often type A. Okay, so influenza A is also known for a propensity for virus reassortment. That's why, okay, it's prone to antigenic changes. Okay, so it likes to rearrange its genes, no, from time to time. Okay, so undergo genetic reassortment. Okay. Now, the subtypes of influenza A that we usually see or that is usually circulating in humans include H1N1 and H3N2. Okay. Now, for influenza type B, okay, they generally change more slowly in terms of their genetic and antigenic properties compared to type A. Okay. Now, there are two lineages for type B. We have the type B Victoria and the type B Yamagata. Okay? Yamagata viruses have circulated much less frequently in comparison to Victoria, no? Globally. Okay? So it's almost always the lineage Victoria that we isolate from human samples. Okay. Okay, now let's discuss the glycoproteins, hemagglutinin and neuraminidase. So hemagglutinin derives its name from its ability to agglutinate erythrocytes under certain conditions. It is known to bind virus particles to their susceptible cells. Okay, so this glycoprotein is also the target of neutralizing antibodies. Okay. Now, the presence of new strains and subsequent influenza epidemics are due to the variability in HA. Okay. So, since there are 18 subtypes, okay, of hemagglutinin, and so the incidence of being able to isolate new strains, it's quite common. Okay. And then hemagglutinin needs to be cleaved. Okay, so it has to be cleaved into its HA1 and HA2 dimers so that the virion can become infectious. Okay, so cleavage must occur so that the attachment can be successful. If hemagglutinin will not undergo cleavage, the virus will not be able to attach onto the receptor sites of susceptible cells. Okay, now for neuraminidase, okay, the glycoprotein neuraminidase is observed to function usually at the end of the viral replication cycle. Okay, so it's a sialidase enzyme that removes sialic acid from glycoconjugates. Okay, so it's very important function is that it facilitates release of the virus particles from infected cell surfaces during the budding process. And it also helps prevent self-aggregation of the virions by removing sialic acid residues from viral glycoproteins. No? So again, its function is usually towards the end. If hemagglutinin functions for attachment, neuraminidase plays a very important role during the removal or separation of the virions from their host cell. Okay, para makagawas from the host cell, it's the enzyme neuraminidase or the glycoprotein neuraminidase is very important. Okay, so what else? It is also possible that neuraminidase can help the virus negotiate through the mucin layer in the respiratory tract. Okay, so it makes it easier for the virions to attack the cells in the respiratory tract because it will allow the viruses to attach to travel through the mucin layer and reach their target epithelial cells. Okay. Now, the viral neuraminidase lowers the viscosity of the mucus film. So, yun yung importansya ng neuraminidase. Okay? So, gaya ng sabi kanina, it helps the virus or the virion negotiate through the respiratory tract. So, basically, it lowers the viscosity of the mucus in the respiratory tract, making it easier for the virion to attach to the epithelial linings. Okay? Okay, so earlier we were talking about antigenic changes, right? So, what are these antigenic changes? So, antigenic changes can be a shift or a drift. Okay, so antigenic changes are quite unique to the influenza virus. They're actually really remarkable because of the frequent antigenic changes that occur in their glycoproteins, no? The hemagglutinin and the neuraminidase. Okay, so what's unique to the influenza virus also is that the antigenic variants, no, or those variants that are created because of the combination or because of the change are actually more advantageous compared to the original strain. Okay? So, ibig sabihin nun, mas lamang yung mga bagong gawa. Okay? Yung mga mix and match ng mga genes compared dun sa original gene or original strain. Okay? So, this makes it difficult for our immune system to create antibodies because they change every now and then, specifically our type A. Okay. Now, what about antigenic shift? Okay. So, antigenic shift is an abrupt change due usually to genetic reassortment with an unrelated strain. So, say for example, si human strain nagmix with swine strain. Okay. So, that will create another type of the virus. Flu virus pa rin, pero it's another version. Okay? Kasi magkaiba yung HA and NA ni human at saka yung HA and NA ni pig. Okay? Ganun din naman if the strain came from a different species, say an aquatic bird or a duck. Okay? It can enter another species, no? Like in this illustration, it's a pig. So, iba rin ang HA and NA ni duck, iba rin ang HA and NA ni pig. So that when the virus from the duck enters the pig, it will undergo reassortment, okay, creating a new strain once it comes out of the pig. Okay? So pag inubo si pig or inatching si pig, lalabas yung ibang strain na naman ng flu virus or influenza virus. Okay? So that's antigenic shift, changing into an entirely different species or strain of the virus. Okay? So the changes in hemagglutinin and neuraminidase usually occur independently. Okay? So pwedeng yung HA lang nag-change, pwedeng yung NA lang nag-change, pwedeng both can change at the same time. Okay? So these changes usually result in the appearance of a new subtype. Okay? And then because it's a new subtype, most likely, pag maraming naapektuhan, it can lead to an epidemic. Okay? Now, let's move on to antigenic drift. Okay, now what about antigenic drift? Okay, so unlike antigenic shift, antigenic drift is described as a gradual change, a minor change in antigenicity due to point mutations that may affect major antigenic sites on the glycoprotein. Okay, so the mutations or the change are usually minor, so it does not create a very remarkable change in the structure. Okay, instead, the changes will only produce viruses that are closely related to one another. And hence, there is still cross protection. So, meaning, taking a look at this illustration or this image here. So, the initial strain or the original strain appears like this. Now, when this virus enters another host, it may undergo point mutation, creating a slight difference. However, if this virus gets into the previous host will be able to defend itself from this new strain because they basically look alike except for a very minor change. So there's still cross protection. Okay. So what else? Okay. Now you might ask, why do these changes happen? Okay. Remember when we described the structure of influenza virus, we said that their genome is segmented. Okay. Now, because of the segmented nature of the genome, when a cell is co-infected by two different viruses of a given type, the mixtures of parental gene segments may be assembled into progeny virions, meaning because of their segmented nature, they're very prone. Excuse me, they're very prone into mixing and matching genomes. Okay, that's why they have this ability to change to create new strains because of their segmented nature. Okay, now this phenomenon we call it genetic reassortment, okay? Mixing and matching of genomes. Okay? So it's called genetic reassortment. It may result in sudden changes in the viral surface antigen, which is a property that explains the immunologic features of the influenza virus. Okay? So this particular feature of an influenza virus, their ability to undergo genetic reassortment, poses a significant problem for vaccine development. You know, that's why we cannot produce a single vaccine that is able to protect us from all types of influenza viruses, you know, because they undergo changes every now and then. And those changes may create new strains that our immune system might not be able to identify with the original strain. So they will look at it differently because of the changes in the antigenicity of the virus. Okay, nagets ba yun? Okay, so hindi nakikilalanin ng immune system natin yung panibagong strain. Okay, kasi nga may change na nangyari. Okay, except of course for minor changes. Okay. So now let's move forward to pathogenesis of our influenza virus. Okay, so the mode of transmission is usually spread from person to person by airborne droplets or by contact with contaminated hands or surfaces. Okay, so usually the incubation period is 1 to 4 days. The infection may occur initially in the respiratory tract. Okay, now, for the viral load or infectivity dosage, usually 3 or a few viral particles can infect an individual already. Okay. So for clinical findings, the influenza virus, of course, causes the respiratory infection influenza or flu, which poses as a serious risk, especially for the very old people and the very young people. Okay. Also, for people with underlying medical conditions, such as those with problems of hearts, in the kidneys, in the lungs, those with diabetes, and also for those with cancers. Okay. Okay, now remember that the virus enters via the respiratory tract in the form of droplets or through things that infected person may use. Okay, so once the virus is inside the body, the viral neuraminidase will lower the viscosity of the mucus film in the respiratory tract. It will, because of this, it will lay bare, okay, the cellular surface receptors, making it easy for the viruses to attack, these receptors and attach to them. Okay, that will promote the spread of the virus faster. Okay. Now, within a short time, many cells in the respiratory tract become infected and they will eventually be destroyed or killed. Okay. Now, the incubation period from exposure to virus and the onset of the illness varies from about 1 day to 4 days, depending upon the size of the viral dose as well as the immune status of the host. Okay, so if a large dose of virus enters into your system, the incubation period will be shorter. Now, in the same manner, if the patient is immune compromised, the incubation period will also be shorter. Okay. Now, viral shedding starts usually the day before the onset of the symptoms. So the day before we see symptoms, that's the time the virus starts to shed. No, they would be shed in the form of respiratory secretions, okay? In phlegm, no? In sipon, yan, okay? In laway, okay? So it peaks within 24 hours and still remain elevated from day 1 to day 2, okay? Eventually, it will decline over a period of 5 days, okay? Now, moving on to signs and symptoms, the clinical manifestations. So the symptoms of influenza usually appear abruptly, and that would include chills, headache, dry cough, okay, or non-productive cough, followed closely by high fever, generalized body pain, muscular aches, okay, malaise, and in some cases, anorexia. Okay, the fever usually lasts from 3 to 5 days. Okay, and it is the same length of time for systemic symptoms. Okay, respiratory symptoms usually last for another 3 to 4 days. Okay, the cough and the weakness may persist for about 2 to 4 weeks. No, luyag yapon, 2 to 4 weeks. Okay, and then of course, some of the major symptoms may subside during the course. Okay, now mild or asymptomatic infections may occur. Okay, so these symptoms may be induced by any strain, pwedeng type A or type B. Okay, in contrast naman, influenza type C rarely causes the influenza syndrome. Okay, usually they cause a form of common cold. Okay, coryza, and then may cough may last for about a week or several weeks. Okay. So for children, ang symptoms nila are quite similar to adults. Although in some children, they have high fever, very high fever, and a higher incidence of gastrointestinal manifestations such as vomiting and diarrhea. Okay, now for the complications, the type A influenza viruses are known as the important cause of croup in children, especially those under 1 year of age. Okay, and then of course, otitis media may develop. Okay, and then pneumonia. Okay, pneumonia is known to complicate influenza. Okay, this can be viral or bacterial or a combination of the two. Okay, so there's increased mucus secretions, which usually leads to the transfer of the agents into the lower respiratory tract. Okay. Now, combined viral and bacterial pneumonia is approximately 3 times more common than primary influenza pneumonia. Okay. Then we have the Reye's syndrome, or you can also read that as Reye's syndrome. It's an acute encephalopathy of children as well as adolescents, usually those between 2 years to 16 years of age. Okay? The mortality rate is very high, about 10 to 40%. Okay? The cause of this syndrome is unknown, but it is recognized as a rare complication of influenza B, influenza A, and then even herpes virus, varicella zoster infections. Okay, so there is a possible relationship between the use of salicylates. So there has been several study linking the use of salicylates and the subsequent development of the Ray's syndrome during the infection. Okay. So for now, the incidence of the syndrome has decreased with the reduced use of salicylates in children with flu-like symptoms. Okay, so now let's move on to laboratory diagnosis and identification. So the specimen of choice for identification of the influenza virus is usually nasal washings or gargles or throat swab. Okay, they're perfect for diagnostic purposes rather than blood sample. Okay, so the collection is best done within the first 3 to 4 days of the infection. So what are the different methods that we can use for identification of the flu virus? So of course, we have the PCR. Okay, so we have RT-PCR. Okay, so they're actually preferred for diagnosis of influenza because of their sensitivity and specificity. Of course, you can do viral culture, either traditional or the shell vial. Okay, in the shell vial, we make use of a very special type of cover slip that we insert into a tube. Okay, you can also use that, or you can go with the traditional using the plates. So viral culture procedures usually take about 3 to 10 days. No, classically we use embryonated eggs or cultures using primary monkey kidney cells. Okay, and then what else? Usually we do not include serum, but definitely trypsin is used. Okay, and then for serology, we have hemagglutination inhibition. Okay, so this procedure usually permits rapid determination of the influenza types and subtypes. Okay, and then you can also perform ELISA. So, and then we have the rapid influenza antigen detection tests. They are commercially available and they're quite easy to use. No? Usually it only takes about 15 minutes. Okay. However, the problem with these types of tests is that they vary in sensitivity and specificity, okay, according to brand. Okay. And then, what else? Okay. So culture, rapid tests, RT-PCR, and serology. Again, those are the methods that we use for identifying flu virus, no? Their types and subtypes. Okay, so for treatment, prevention, and control, um, if the infection is already existing, a treatment is usually through the use of drugs. Okay, so we have two groups. We have the adamantine drugs, which are basically M2 channel inhibitors. We use this widely for treatment of influenza type A infections. Included under this type are the amantadine hydrochloride and rimantadine drugs. Okay? Now, apart from the amantadine drugs, we can also go for neuraminidase inhibitors. Okay? So neuraminidase inhibitors include zanamivir and oseltamivir. So these are actually common drugs, no? Antiviral drugs. So zanamivir and oseltamivir. These are neuraminidase inhibitors. So for NA inhibitors, they are widely used to treat both type A and type B infections. Okay. Now for prevention and control with the use of vaccines, okay, inactivated viral vaccines are the primary means of preventing influenza. Okay, the currently existing vaccines are continually being rendered obsolete, okay, because remember, our viruses undergo antigenic drift and shift. Okay, so the existing vaccines may be helpful now, but they will be rendered useless, okay, when new strains are already created. Okay, now subtypes of influenza circulating around the world are constantly being monitored so that we could create vaccines for those strains. Okay, so vaccine types are inactivated viral vaccines, live virus vaccines, and influenza vaccines. Remember, flu vaccines are given yearly. Okay, so we have to get them yearly. We have to get the shots yearly also. Okay. Uh, Hand transmission is also potentially important. That's why we can prevent a lot of infections by practicing good hand hygiene. Okay, so practice hand washing with soap and water, and you can also use alcohol-based hand rubs. They're highly effective. Of course, your hands should be free from physical dirt. No, so if your hands look clean, you can use alcohol-based hand rubs. But if they are soiled, no, and if your hands are really dirty, it's best to wash them with soap and water. Okay, and that ends our discussion on influenza virus. Thank you for listening and watching. Rewatch if you need and learn some more by reading the particular chapter for influenza virus in your Jowets.